Usually when you think about your own survival, you focus on the absolute non-negotiable necessities: breathing, eating, drinking water.
Right, the really basic stuff.
Exactly. If you stop doing any of those things, the individual organism, whether that is you listening right now or me or a goldfish, is just entirely done for.
Yeah, it's completely binary.
You live, you don't, you die.
The rules of individual survival really are ruthlessly straightforward. I mean, every calorie you consume, every breath you take is entirely dedicated to maintaining your own biological machinery.
But then you look at biological reproduction, and suddenly that fundamental ruthless rule book is thrown completely out the window.
Oh, totally. It makes no sense from that perspective.
Right. We are looking at a physiological process that takes an astronomical amount of energy, yet is utterly unnecessary for you, the individual, to stay alive.
It is the ultimate biological paradox. Why burn precious calories building a new organism when you could use that energy to run away from a predator or just survive a harsh winter?
Welcome to the deep dive. Today we are unpacking a really fascinating stack of scientific source material detailing the exact mechanics of biological reproduction.
It's a great topic. Glad to be here to break it down.
Our mission today is to explore this exact paradox of why life bothers to create more life, and to trace the incredible biological engineering that scales from microscopic, single-celled organisms right up to the intricate complexities of human reproductive health.
It's quite the journey.
It really is. Okay, let's unpack this. Why does an organism waste so much precious energy on a process it absolutely does not need to survive?
Well, because if we look past the individual, the answer becomes pretty obvious. If a single non-reproducing organism existed out there in the wild, you and I wouldn't even notice it.
Because there's only one.
Exactly. We only notice a species because there are massive numbers of them. So while reproduction is a terrible energy investment for the individual, without it, the whole lineage just ends.
So the energy is spent not for the self, but for the continuation of the species as a whole.
Spot on. And to continue that lineage, you have to pass down the instructions. Our sources bring us down to the most basic fundamental event in all of reproduction, which is copying the blueprint.
We are talking about DNA.
Yes, the deoxyribonucleic acid sitting inside the nucleus of a cell. It carries the exact architectural instructions for making proteins and, well, designing the whole body.
So how does a cell actually handle that blueprint?
Well, before a cell can even think about dividing, it must create a perfect or near-perfect copy of that DNA. But it can't just spit the raw, naked DNA out of the nucleus and hope for the best.
Right, that wouldn't end well.
No, a raw string of DNA without a cellular structure around it to maintain life processes would immediately degrade and die. Therefore, copying the DNA has to be accompanied by the creation of an entirely new cellular apparatus.
Like a membrane organelles the work.
Exactly. Only then can the DNA copies separate, with each taking its own fully functioning life support system with it.
Here's where it gets really interesting, though. The chemical reactions that cells use to copy this DNA are never absolutely 100% reliable.
No, they aren't.
It's basically like having a slightly glitchy photocopier. Every time you hit print, the copy is incredibly similar to the original, but it is not completely identical. There is always a tiny smudge or a misprinted letter, a slight variation.
And what's fascinating here is that these tiny glitches, these subtle variations in the copying process, are the entire basis for biological evolution.
Wait, really? The mistakes are the basis for evolution.
Yeah. I mean, a lot of the time, the variations are so drastic that the new DNA can't work with the cellular apparatus at all, and that newborn cell just dies.
Oh, wow.
But those surviving, subtly different cells are the key to a species surviving a changing world.
Our sources give a really good example of this. Imagine a population of bacteria living happily in a temperate pool of water.
Right, a nice, comfortable environment.
Yeah, they are perfectly suited to that specific temperature. But then, global warming or a geothermal shift drastically raises the water temperature.
In that scenario, the vast majority of those bacteria are going to be wiped out. Their standard blueprint simply isn't built for the heat. Their proteins will denature.
They just cook.
Pretty much. However, because of that glitchy photocopier effect, a few variants in the population might have a random mutation, a glitch, that accidentally makes their cellular walls more heat resistant.
And those are the ones that survive.
Exactly. Those few survivors will live, multiply, and repopulate the waters. The random variation protected the species, even though the original individuals perished.
So understanding the DNA must be copied and physically separated with its own machinery. How do simple organisms actually execute this on their own? If an organism is just a single cell, do they basically just hit print to make a copy of themselves?
Kind of, yeah.
How does that physically work in three-dimensional space?
For single-celled organisms, the process is called fission, and the physical mechanics depend heavily on the organism's architectural design. Take the amoeba, for instance.
The classic microscopic blob.
Right, it's essentially a fluid blob. Because it lacks a rigid, asymmetrical structure, the splitting of its cell into two equal halves can take place in absolutely any plane. It literally just pinches itself anywhere in the middle and pulls apart.
But not all single-celled creatures are just blobs.
No, they definitely aren't.
Like, take leishmania, the organism responsible for the disease Kala-Azar, which our sources note, is a really severe fever-inducing illness transmitted by sandflies.
Nasty stuff.
If you look at leishmania under a microscope, it isn't a blob. It has this long whip-like structure at one end called a flagellum, which it uses to move.
And because of that distinct structural orientation, binary fission has to occur in a very definite, specific longitudinal plane.
So it has rules.
Exactly. It has to split right down the middle in relation to that whip, ensuring both new cells get the correct foundational structures to grow their own whip. It can't just pinch arbitrarily like the amoeba, or one daughter cell would be fundamentally deformed.
And then you have the malarial parasite Plasmodium. It completely ignores the divide into two rule.
Oh yeah, Plasmodium is wild.
It performs multiple fission, it builds a protective cyst wall around itself, and inside that wall, the nucleus divides into many, many daughter cells simultaneously.
And then when the conditions are right, the wall bursts open and a whole swarm of parasites is released at once.
It is a brilliant, if terrifying, workaround. And nature's hacks scale up to simple multicellular organisms, too.
They do.
Like Spirogyra, a type of green algae you might see as pond scum, basically just fragments. As the filament matures, it simply breaks into smaller pieces, and each fragment just grows into a new, fully formed individual.
And the regenerative capabilities of some simple animals are even wilder. Organisms like the hydra or planaria, which are flatworms, can be physically chopped into multiple pieces.
Which sounds fatal.
You'd think so. But specialized cells in every single one of those severed pieces will rapidly proliferate. They form a mass of cells that then undergo highly organized changes to regenerate into entirely complete, separate flatworms.
That is insane.
The hydra can also just grow a tiny version of itself as a bud right on its side, which eventually pops off as an independent individual.
Okay, but I have to push back here.
Sure.
If regeneration is so incredibly easy and efficient for a flatworm, why don't complex animals do it? If I accidentally cut my finger off chopping vegetables, the wound heals, but the severed finger doesn't grow a second, identical me.
No.
Why the limitation?
That's a great question. It really comes down to architectural complexity and tissue specialization. In a flatworm, the cells are relatively adaptable.
Okay.
But in your body, you aren't just dealing with a random collection of cells. Your cells are locked into highly specialized roles. They form specific tissues like muscle or nerve.
Right.
And those tissues form complex organs, which must be placed at exact, precise positions in a closed circulatory and nervous system. A simple cell-by-cell division, or trying to sprout a whole human heart and brain from the tissue of a severed finger, is biologically impossible.
Because the cells in the finger don't have the big picture instructions anymore.
Exactly. They no longer have the active instructions or the systemic support to build a brain.
But farmers and gardeners somehow manage to cheat this limitation with complex plants. You can't grow a human from a finger, but you can absolutely grow a new rosebush from a single cut stem.
Yes, plants are a special case. They utilize vegetative propagation. They possess specialized areas of actively dividing unspecialized cells, often in their roots, stems, or leaves.
Which act kind of like blank slates.
Precisely. They retain the ability to develop into entirely new plants given the right conditions, like moist soil.
Which is a massive advantage for agriculture. Our texts note it allows us to propagate plants like bananas, oranges, and certain roses that have actually lost the capacity to produce viable seeds entirely.
Yeah, if you eat a commercial banana, you aren't planting seeds to get more bananas.
Right. Farmers rely purely on this cloning method. Plus, every new plant is an exact genetic clone of the parent, which locks in the specific taste and traits we want.
Fungi have their own solo method, too. Think of rhizopus, the common bread mold.
Oh, yeah, the fuzzy stuff.
Exactly. If you leave wet bread in a dark cupboard, you get those fuzzy, thread-like structures. But the actual reproduction happens in these microscopic blobs on stalks called sporangia.
And they contain spores, right?
Yes, wrapped in thick protective walls. Those spores just wait, sometimes floating in the air for ages, until they hit a moist, nutrient-rich surface like a slice of bread.
And boom, the wall dissolves and they start growing.
Exactly. But notice the recurring theme across all these examples. Asexual reproduction, whether it's an amoeba splitting, an algae fragmenting, a hydra budding, or a mold releasing spores, relies on a single individual.
Right.
And while it is incredibly fast and efficient, it brings us right back to our glitchy photocopier.
Because if you are just copying yourself over and over, variation is agonizingly slow. You're relying entirely on random, tiny copying errors to build up over thousands of generations to survive a changing environment.
Which is very risky.
Which leads us to the big evolutionary pivot. If going solo is so fast and energy efficient, why did nature invent a system that requires two totally separate individuals to find each other and cooperate?
It is entirely about evolutionary math. Asexual variation is simply too slow in a rapidly changing environment.
So the clones are too similar.
Exactly. If a new disease or climate shift appears, a population of clones might be entirely wiped out before a useful mutation naturally occurs. But if you take variations from two completely different individuals and combine them, you instantly create a novel, never-before-seen combination of DNA.
It safely and drastically speeds up the creation of variants, giving the species a massive survival toolkit.
That's it, exactly.
It's like taking two massive IKEA instruction manuals, both with their own unique edits, scribbles, and notes, and merging them together to build a completely new piece of furniture.
That's a great way to look at it.
But wait, hold on. The math doesn't actually work. If you combine two full blueprints from two different individuals, wouldn't every new generation end up with double the amount of DNA?
Oh, I see where you're going with this.
Yeah. Generation 1 has 100%, Generation 2 has 200%, Generation 3 has 400%. The nucleus would explode. That sounds like a biological disaster.
You are absolutely right. A cell cannot survive with double the operating instructions. It would be chaos. So, to solve this exact math problem, complex organisms developed a highly specialized process called meiosis.
Meiosis?
Yes. In specific reproductive organs, certain lineages of cells undergo a unique type of cell division that purposefully halves the number of chromosomes.
Wait, how does a cell function on half the instructions without dying? You just said a cell needs its blueprint.
Well, these special cells, germ cells, aren't functioning skin or liver cells. They're essentially stripped-down delivery vehicles. They don't need to perform daily life functions.
So they're just carrying cargo.
Precisely. Their only job is to carry half the blueprint and survive long enough to meet another germ cell. That way, when a germ cell from individual A meets a germ cell from individual B, they fuse.
And the math balances out.
Exactly. The resulting zygote now has the perfectly correct, reestablished 100% amount of normal DNA, half from each parent.
And as organisms get more complex, those half DNA germ cells specialize their roles. One germ cell becomes very large to store a massive amount of food and energy to power the initial growth of the new organism.
Which we call the female gamete, or the egg.
Right. And the other germ cell shrinks down, sheds excess weight, and becomes highly motile. It is basically a microscopic torpedo with a tail, optimized purely to seek out the egg.
And that's the male gamete, or sperm. You see this dynamic beautifully illustrated in flowering plants, actually.
Our sources go into detail on this.
They do. The flower itself is just a flashy reproductive center designed to get these gametes together. The male part, the stamen, produces yellowish pollen grains.
And the female part?
The pistil, which sits right in the center and houses the ovary at its swollen base.
The journey the pollen takes is genuinely incredible. A grain of pollen gets blown by the wind or carried by an animal, and it lands on the sticky top of the pistil, called the stigma.
But that's just the roof of the building. The egg is down in the basement.
Exactly. So the pollen grain literally has to chemically bore a tunnel. It grows a microscopic tube all the way down through the stalk of the pistil to reach the female germ cells hidden inside the ovary.
It's quite the engineering feat.
And once the male genetic payload travels down that tube and fertilization happens, the ovule develops a tough coat and becomes a seed. Meanwhile, the tissue of the ovary surrounding it miraculously swells, changes its chemical composition, and ripens into a fruit.
And this absolute requirement for highly specialized reproductive organs to create, house, and deliver these half-DNA germ cells leads us directly to how your own species handles the process.
Humans use this exact same sexual mode of reproduction, but our timeline is uniquely paced. We do not exit the womb ready to reproduce.
No, and for very good reason. Early in a human's life, all of the body's massive energy resources are entirely directed towards general somatic growth.
Like getting taller, acquiring adult teeth, expanding lung capacity.
Building brain mass. It is a massive energy drain just to build the adult frame. It is only as that rapid rate of general physical growth begins to slow down that the body shifts its hormonal priorities.
And the maturation of reproductive tissue finally takes center stage.
Yes, this transitional period is puberty.
And it is a notoriously slow, often awkward transition. Our sources note the physical markers. There are common changes we all know, like new hair growth in the armpits and genital areas, and the skin becoming oilier, sometimes leading to acne.
The fun stuff.
Right. Then there are specific changes. Boys developing thicker facial hair and their vocal cords enlarging so their voices crack and deepen, while girls experience breast enlargement and the beginning of the menstrual cycle. But why does this have to happen so slowly? And why is it so highly visible?
If we connect this to the bigger picture, animals that mate internally need a biological way to broadcast their state of sexual maturity to other individuals.
So it's basically advertising.
Essentially, yes. These physical changes, the hair growth patterns, the changes in body proportions, the scent changes from oil glands, are chemical and visual billboards. They are biological signals indicating that the specialized germ cells are now actively being produced, and the individual is physically capable of reproduction.
Let's look at the actual biological engineering behind those human germ cells, starting with the male anatomy. The formation of sperm takes place in the testes,
Right.
but incredibly, they are located entirely outside the main abdominal cavity, housed in a pouch called the scrotum. Why put such a vital organ in such a vulnerable external position?
Because the creation of sperm is highly sensitive to heat. It requires a temperature slightly lower than your normal core body temperature.
Oh, so if the testes were inside the warm torso.
The glitchy photocopier wouldn't work at all. The sperm would fail to develop.
Got it.
Once formed in that cooler environment, the sperm travel up into the body through a tube called the vas deferens. Along this highway, glands like the prostate and the seminal vesicles add specialized fluids.
So it's not just liquid for transport.
No, it is a complex cocktail providing crucial nutrition, mostly fructose, for the sperm. Remember, the sperm are basically just dense genetic payloads attached to a highly motile tail. They need external fuel to make their journey.
Then you have the female anatomy. At birth, a human female's ovaries already contain thousands of immature eggs. They are just waiting.
For years.
Yes, until puberty hits and a biological clock starts. About once a month, a hormone signal causes one of those eggs to mature and be released. It travels down the fallopian tube, making its way toward the uterus, which is a highly elastic, muscular bag-like structure.
And if that egg happens to encounter sperm in the fallopian tube and fertilization successfully occurs, the resulting embryo travels down and implants itself into the thick lining of the uterus.
And this is where we see one of the most stunning pieces of biological engineering in the mammalian world, the placenta.
The placenta is wild.
It sounds like an intense biological border control and trade port. It is a specialized disc of tissue embedded directly in the uterine wall. On the embryo side of this tissue, it is covered in these tiny, finger-like projections called villi.
And on the mother's side, there are massive blood spaces that entirely surround those villi.
Wait, so the mother's blood and the baby's blood mix?
Actually, no. They never actually mix. That specific Physical design, thousands of tiny fingers dipping into pools of blood, creates an absolutely massive surface area.
Ah, so they just get really close.
Exactly. This surface area is required to diffuse molecules right across the barrier. It allows vital glucose and oxygen from the mother's bloodstream to pass efficiently into the developing embryo.
And I imagine it goes the other way too.
Yes, simultaneously taking the toxic waste substances generated by the baby and transferring them back into the mother's blood where her kidneys can clear them out. It is a perfect, temporary, biomechanical life support system that lasts for 9 months.
But that raises the obvious question. What happens when fertilization doesn't occur? Because the female biological system operates on a continuous monthly loop.
Right, it's constantly preparing for a pregnancy that the vast majority of the time does not happen.
So what does this all mean when the egg isn't fertilized?
Well, when an egg is released but not fertilized, it only survives for about a day. But the uterus had already spent weeks aggressively preparing itself, building up a thick, spongy inner lining, richly supplied with blood vessels to nourish a potential embryo.
Which is now useless.
Exactly. When the hormone levels drop, signaling that no embryo has implanted, that lining is no longer needed. So the tissue slowly breaks down. It exits the body through the vagina as blood and mucus every month. This cycle is menstruation.
Which brings up a really crucial distinction highlighted in our sources.
Yeah.
Just because a human body has undergone puberty, produces gametes, and is physically capable of reproduction, absolutely does not mean the mind or the individual is ready for that massive, life-altering responsibility.
Human reproduction isn't purely a biological function. It is deeply entangled with our social, psychological, and economic reality.
Individuals face immense pressures from all sides.
They really do. Friends might exert peer pressure regarding sexual activity before someone is mentally ready. Families often exert heavy cultural pressure to get married and produce heirs.
Conversely, governments or harsh economic conditions might exert pressure to strictly avoid having children.
Navigating those choices is incredibly complex. Consequently, humans have developed tools to separate the intimacy of the sexual act from both the biological result of reproduction and the transmission of disease.
Right, because the sexual act is a very intimate connection of bodily fluids and tissues. It's a prime vector for sexually transmitted diseases. Our sources specifically categorize these STDs.
Yes, they break them down into bacterial infections like gonorrhea and syphilis and viral infections such as warts and HIV/AIDS.
And preventing the transmission of these diseases, while also managing reproductive choices, has led to various sophisticated methods of contraception.
We can break these solutions down into 4 distinct categories. First, you have mechanical barriers like condoms.
Which are quite literally physical walls.
Exactly. They stop sperm from ever reaching the egg. Importantly, because they block skin-to-skin and fluid contact, they are the primary method for preventing the spread of the STDs we just mentioned.
Second, our text outlines hormonal methods. These are typically oral pills, and they work by tricking the body's internal chemistry.
They alter the hormone balance so the ovaries simply never release an egg in the first place. No egg, no fertilization.
But because they are fundamentally altering your body's natural endocrine system, they can carry various side effects.
Third, there are intrauterine devices, like the copper T-loop. This is a small device placed directly inside the uterus. The copper ions naturally released by the device create a chemically toxic environment for sperm, paralyzing them and preventing a pregnancy from taking hold.
Though the texts note the physical presence of the loop can sometimes cause uterine irritation.
Right. And finally, there are surgical methods. In males, the vas deferens, that highway we talked about, can be physically cut and blocked, so sperm is never transferred out of the testes.
And in females, the fallopian tubes can be surgically blocked, so the egg can never reach the uterus and the sperm can never reach the egg. While surgical methods are highly effective, safe, long-term solutions for preventing pregnancy, our source material highlights a really crucial warning about how surgical interventions are sometimes misused.
Yes, with profound societal consequences.
Specifically, the reckless misuse of surgery for illegal sex-selective abortion, or female feticide. When people use ultrasound technology to determine the sex of a fetus and then use surgery to selectively terminate female pregnancies due to cultural preferences for male heirs, It disastrously skews the male-to-female sex ratio in those societies.
It is a stark example of how a biological tool, when applied with cultural bias, creates a massive demographic imbalance.
An imbalance that threatens the stability of a society.
This raises an important question, actually, one that our sources pose regarding the broader human population.
Go ahead.
When discussing reproduction, it is often argued that an endlessly expanding human population makes it increasingly difficult to improve the standard of living for everyone, as resources are stretched thin.
That's a common argument.
But we are challenged by the text to look deeper at the mechanics of society. Is a raw population number the true issue? Or is severe social inequality and the uneven distribution of resources actually the primary reason for poor living standards?
It is a heavy thought, and it really shows the incredible scope of what we've unpacked today. We started with the microscopic mechanics of a single cell.
We looked at how a glitchy DNA photocopier is the invisible engine driving all of evolution.
We marveled at the rapid cloning hacks of flatworms and the seedless vegetative propagation of our favorite fruits.
And we traced that foundational biology all the way up to the immense complexity of the human body. From the delayed, socially broadcast onset of puberty to the highly specialized organs delivering half strands of DNA.
To the intricate engineering of the placenta and the continuous monthly reset of the menstrual cycle. It is an incredible biological journey.
It really is.
But before we wrap up this deep dive, I will leave you with a final thought to mull over, building on everything we've discussed today.
Let's hear it.
We established at the very beginning that the entire foundation of biological evolution relies on accumulated genetic variations, those tiny random glitches passed down organically from one generation to the next through the sheer chaos of reproduction.
Right, nature's glitchy photocopier.
But look at humans now. We are using technology to completely alter that landscape. With hormonal pills, surgical blocks, tissue cultures, and advanced medical interventions, we have the unprecedented ability to completely control, pause, or artificially select exactly how and when reproduction happens.
That's very true. We're stepping out of the organic loop.
So, if evolution is driven by the organic, slightly chaotic passing of the blueprint, how might our modern ability to essentially rewrite the rules of the biological mating game alter the future evolutionary blueprint of the human species?